Okay, let's get started. Seems like it's been a long time since you were here, but I know it was only last Thursday. I hope you all had a chance to do something fun over the weekend after a stressful maybe first week. I wasn't maybe super clear about that first homework assignment, so I might have mentioned it last time, I forget. There is a little 2-question introductory homework assignment that I have set to be due this Friday at midnight. So I looked this morning, 120 out of 140 of you do have access. You're in my grade book. If you are still having trouble getting logged in, or know that you won't be able to purchase that for a while yet, do talk to me. But again, it's just 2 questions, out of like 500 total that you have. But that's just to encourage you, hey, let's get in, make sure you're getting started on that. The rest of your homework is not due till the first exam, which is another couple weeks away. Okay. So last time we finished, we're finished with chapter 1. We finished with last time that little piece of chapter 6 where we talked about culturing bacteria in the lab. We finished that talking about some different medias. We talked about principles of microscopy into chapter 4. Also started talking taxonomy a little bit. So where we left off, we were thinking through some of the contributions of Linnaeus. He's the one that suggested the binomial nomenclature, that every organism needs to have 2 names, that it needs to be in the language of science, which is Latin. And we talked about how to write those names grammatically correct. And we just kind of discussed a little bit how important it is to be organized in what we know about organisms, so that there's no confusion that I'm talking about one organism and somebody somewhere else is actually talking about a different organism when we think we're talking about the same thing. So especially as new species are identified or new viral strains, we kind of, again, we want to be organized in what we know. Okay, so kind of continue and to kind of finish up talking about taxonomy. Linnaeus proposed only 2 different kingdoms. So I wrote over to the side here. Remember, Linnaeus is best known and kind of organizing the information that we do know and setting up this hierarchical system. He proposed 2 different kingdoms, plants and animals. Okay, we've been revising it ever since. And certainly scientists don't always agree on how we should group organisms or even what we should name them. We have to kind of do the best we can, I guess. When I was in high school, in the 80s, we learned a 5-kingdom system. So that was the Whittaker system that was developed in 1969. Those 5 kingdoms were animals, plants, fungi. Plants and animals until fairly recently before that were considered in the same kingdom. But there's one very big thing that separates plants and fungi. Any ideas what that might be? It's something that plants do that fungi don't. photosynthesize, right? So that's kind of the big difference between those two kingdoms, or what we used to call kingdoms, protozoa or protists, either way. And Monera is an old-fashioned term. We don't use it anymore, but that was where we put all the bacteria in this kingdom we called Monera. Okay. Now, so we always used kind of shared characteristics to group organisms into different groups, and we still rely on that to some extent. So we're going to talk about what some of those characteristics are shortly, and what we have used to classify organisms. But today we're putting a much bigger emphasis on genetic comparisons and more molecular means to put organisms into similar groups, because these genetic comparisons take into account phylogenetic relationships. Okay, that's a fancy way to say evolutionary relationships. Okay, so phylogeny refers to evolutionary relationships, meaning what was the common ancestor? So the thinking is that if two organisms had a common ancestor not that long ago, that then they are more closely related to organisms that had a common ancestor much earlier. Okay, so kind of a practical application of phylogeny is that you may have heard the debate, in the media about SARS-CoV-2 and where it came from. Okay, so there are people that are trying to figure out exactly when the coronavirus that we're dealing with right now mutated, or what we would say spilled over. So we think that this is probably a virus that originally infected some animal, probably bats. So at what point did that virus suddenly be able to infect animals or other animals and not just bats? We call that a spillover event. The interesting article, and I wish I would have taken a screenshot of this, but it was an opinion piece in the Wall Street Journal in August, but it was opinion of a famous virologist that used to be the director of the CDC. His name is Robert Redfield. Okay, but the article's called "The World Needs to Know What Happened at the Wuhan Lab." All right, so you might have heard there's this debate. Well, was this really a virus that got accidentally released from a lab? And at first, that was poo-pooed on. That can happen. But the Chinese have been very secretive about that and letting people go in and investigate. They were doing gain of function research at that lab on coronaviruses. In other words, they were trying to make a coronavirus that could do things that it couldn't do before, like infect people. That's what they were trying to do. And they were getting funding from a lab in the US. So if that is, really what happened, that it was a virus that escaped, we kind of need to know that, right? I think there's good evidence that we shouldn't be doing those kind of experiments, certainly. But it's all sort of circumstantial evidence at this point. The evidence that they look for in tracing the evolution of a virus is not there like it should be if it came from an animal. So it's maybe one of those things that we never know for sure, but there's a lot of people that are really good at tracing the evolution of animals, the phylogeny. All right, so just a little tangent there, perhaps. So like I said, we're putting more emphasis on looking at genetic comparisons, especially, and that's primarily due to the work of Carl Woese. And sometimes we give credit to George Fox. He was in Carl Woese's laboratory. This is some work that they did in the 1970s, where they were sequencing, they sequenced the ribosomal RNA of many organisms. And at that time, sequencing was a really laborious process. Like they had to run gels They had to do all this by hand. It took a really long time, not like what it does now. But they were sequencing the ribosomal RNA of many animals to try to understand their evolutionary relationships. Why did they use ribosomal RNA? And well, what is ribosomal RNA? We're going to talk more about ribosomes shortly. We're talking about structures of cells, but again, later on, we're talking about transcription and translation. But ribosomal RNA is the kind of RNA that you find in ribosomes. Ribosomal RNA and protein is what makes up ribosomes, which of course is where proteins are manufactured in the cell, both bacterial cells and eukaryotic cells. So they were looking at ribosomal RNA because ribosomal RNA doesn't tend to mutate very quickly. All right, if it does mutate, then most likely the organism will die, because if the organism's not making the correct proteins, then it usually won't survive. So we say ribosomal RNA is highly conserved, meaning it has a really slow mutation rate. Okay, so that's why you chose that molecule to sequence instead of DNA. And what they expected to find, they expected all of life to be able to have like two basic groups. Okay, he expected the ribosomal RNA for prokaryotes to look different than the ribosomal RNA of eukaryotes. Surprisingly, though, that's not what he found. He found that all of life kind of fit really nicely into three groups and not two. Okay, so this is a really, one of those mind-blowing things. So he came out with a new taxonomic scheme in the early 1990s. And he said that there should be another level above kingdom, above kingdom, that he called domain. Okay, so he advocated there should be three domains of life: the bacteria, the archaea, and then the eukarya. Okay, so if we go back to our kind of hierarchy here, he said, okay, let's put something above kingdom here, domain. Okay, and that's the accepted hierarchy now. It may not be as recognizable as those other levels. So I'm surprised that your book doesn't have Wose's tree of life. Okay, I got this off the internet, but I didn't even cite it because it's just pretty much well known now. But here's the three domains that he suggested. You are here, all right? We're animals, so we belong in the eukarya. All of these, this is not to memorize, but all of these other taxa are correctly called kingdoms now. Again, that's not generally recognizable to a lot of people. A lot of these over here in the eukarya are what we would used to call protozoa, protists. And we'll talk more about those in the coming week or so. Archaea, remember the archaea and the bacteria are both considered prokaryotic cells. We think about the archaea as being kind of extreme organisms, but that's not necessarily true. There are archaea that live in your body right now, most likely. But there are some that live in very extreme environments, like halophiles. They live best in high salt environments, or this Thermoproteus or pyrodictum. Pyro makes you think fire or heat. So they live in warmer temperatures, ideally. Prefer those warmer temperatures. There's organisms that make methane, right? Those are all just kingdoms in that archaea group. And then even a lot of the bacteria are not really recognizable, even to me that likes to read about these sorts of things. We're going to talk about what it means to be gram-positive or gram-negative, but a lot of the bacteria that we study are going to be in that gram-positive group, or this proteobacteria group, which includes a lot of gram-negative. But there's some photosynthetic bacteria. Okay, so that's the phylogenetic tree of life. One of the really exciting or interesting things that he found when he looked at the common ancestry is that the archaea, we share a closer common ancestor with the archaea, which you would think that the bacteria and the archaea would be off on one side, and they would share a closer common ancestor than with the eukaryotes. So that was sort of surprising. And it kind of reflects that, you know, when we look at shared characteristics, well, sometimes the archaea have things more like the eukaryotes, sometimes they're more like the prokaryotes, and sometimes they just have something that's totally unique. Right? One other question, what about viruses? Where do you think viruses would fall in this scheme? Kind of a trick question. Remember, this is based on ribosomal RNA sequencing. Actually, viruses don't have ribosomes, and we haven't talked about them yet. So they're not actually on this list, and most often are really not considered alive. There's some disagreement about that in the scientific community, but mostly it's agreed that they don't satisfy the characteristics that you would need to have to be considered alive. So we'll talk more about that later. Just wanted to throw that in there. All right, so that kind of leads us into thinking about what kind of characteristics we use to classify organisms, talk a little bit more about the ones that we can easily see, or maybe say easily with a microscope anyway. But chapter 11, we're just going to dip into just a little bit, just kind of a few pages of that, where it's talking about general characteristics of prokaryotic organisms. So right at the beginning there, they talk, well, what is the difference between a bacteria and an archaea? There are, and I thought later, well, I thought a few days ago, I should have eukarya, might as well compare all three of them, but in your book, this is the way they do that. So just kind of picking out a few characteristics that are different between the bacteria and the archaea. The big one that we always kind of seem to come back to is the cell wall structure. And we're going to talk a little bit more closely about that. I think we'll get to it today. What is peptidoglycan exactly? We're going to talk more about that, but that is a compound that any bacteria that has a cell wall is going to have peptidoglycan in it. Archaea do not have peptidoglycan. They have something kind of similar sometimes. There's a lot of variation there, but it's not the same thing. And of course, any eukaryote that has a cell wall, like a plant or an animal, or not an animal, a plant or a fungi, doesn't ever have peptidoglycan. So that's something that's really unique to bacteria. And then just kind of picking out a couple of other things. We don't talk at length about membrane lipid composition, kind of leave that more for your biochemistry classes. But basically, the bacteria have membranes that are kind of similar to ours in a lot of ways, but the archaea have membrane structure that's kind of unique to them. Okay, so I'm not worried about the details there. A lot of archaea have metabolisms that are similar to bacteria, but then the genetics is kind of more like the eukarya sometimes. So for example, they have an AUG start codon that encodes for methionine, like eukaryotes. We'll get more into that later, but bacteria always start out their proteins with the amino acid formylmethionine. So I don't know, just a smattering of, you know, a few characteristics that are different between those. Okay, but mostly we're going to focus on bacteria and their characteristics. Kind of cut off my title there. but it was the same as the previous page. Okay, so kind of microbiology 101. What do cells look like? This is an easy way that we can group organisms together. And again, if you're in lab, you'll get to see some of the diversity, well, not a lot of diversity, but you'll get to see a little bit. Cells that are round, we have a special name for them. We call them coccus, a coccus. Coccus, similar, cocci, sorry, coccus singular, cocci plural. That just means a round cell. And then this is the other most common shape or morphology of a cell, a rod shaped, or we call that a bacillus. Okay, so if you're in lab, you're going to see lots of cocci, lots of bacilli. Probably won't get a chance to view any other morphology, but there's a lot of other stuff out there. There's some organisms that are kind of not perfectly round, but not really considered a rod either. So somewhere between those. So we call that a coccobacillus. There's some stranger stuff like comma-shaped cells. We call those Vibrio. The causative agent of cholera is a Vibrio. There are helical shaped organisms, the spirillum and the spirochetes. Let me come back to those in a minute because those are not as easy to tell apart. Some organisms are considered pleomorphic. That means they have no set shape. And it may be that they have a different shape depending on kind of their environment, but cells that don't have a cell wall, there are a few bacteria that actually don't have a cell wall, and those can be kind of any shape, pleomorphic. And then there's just some other kind of bizarre stuff like star-shaped or even square. So there's some kind of unique things out there. But how do we tell a difference between a spirillum and a spirochete? Both of these organisms have morphologies that allow them to move like a corkscrew through medium. So maybe if they're in a medium that's a little more thick, a little bit more viscous, this would enable them to move through that towards whatever they're trying to move towards. The spirillum is a little bit more rigid, meaning it just kind of flexes just a little bit when it moves through, while the spirochetes are much more flexible. Oftentimes, although you can't see any flagella in this picture, oftentimes the spirillum will have flagella at the ends. We'll talk more about flagella shortly. That's what makes them able to move through the medium. And the spirochetes have a special kind of internal flagella that's fairly unique. Oops, flagella. So that's pictured down there. It's basically enclosed in a membrane here. You can kind of see it right here. And that's just a little specialized kind of flagellum. Okay, so here's another picture that compares those two. Thinking about what kind of microscopy these pictures are. Well, good. Sometimes pictures aren't labeled very well, what kind of microscopy they are. Like these. The one on the right, that's a spirillum. What kind of microscopy do you think that is? Maybe hard to tell when I don't give you the units. But that you can see kind of the, you can really see the texture on the actual cells. Okay, that's not light microscopy. Okay, it's actually a scanning electron micrograph. Remember the difference between the scanning and the transmission micrograph. The transmission is always going to be a cross section. Okay, so this is definitely a 3D image. You can see the whole cell. Those little brown things are the flagella. So they added the color into that later. It's not like stained with particular dyes. It's just artificially added color. Okay, so that's spirillum. That is actually, that organism is Helicobacter pylori. So that's the organism that we now know causes a large portion of gastric ulcers, stomach ulcers. Okay, and then the picture on the right, from looking at that with that dark background, you know that it's either dark field microscopy or fluorescent microscopy. Okay, and I'm 99.9% sure that this is dark field micrograph, because usually that the reason that we do dark field is that so we can see organisms moving around, because remember, we don't need to stain them. Okay, and spirillum are really cool to see moving around. That's probably why you would want to look at them at all, just to see that spiral movement. So I'm pretty sure that's dark field. It's hard to tell if that's actually, fluorescing. Like, I don't think that usually fluorescent dyes are either like green or red or yellow. So that's actually the organism that causes Lyme disease, Borrelia burgdorferi. Okay, that's another example of that's a spirochete. Okay, and this YouTube video, take a look at that sometime. That's a dark field micrograph showing those spirochetes moving around. Okay, one of the disadvantages of me recording the lectures are that I can't easily just show you an animation. I can't. So take a peek at that on your own time. It's kind of cool to be able to watch bacteria actually moving around. Okay, and the spirochetes, again, they're just a little bit more flexible. They kind of just that whole body moves around a little bit in a little bit different way. All right, so that's the shape or morphology of cells. Another way that we can categorize organisms is the arrangement of the cells themselves. All right, so the arrangement is going to be based on how many planes of division in other words, does it only divide? So when bacteria cells divide, one cell becomes two, right? So does it always split in the same spot, or can it split, crosswise or diagonally or another way? Okay. The other thing that is in consideration here is, do the cells stay attached? Okay, that's the other thing that will contribute to how the cells arrange themselves. Typically, so if we look at cells under the microscope, this can help us identify organisms, because one species will typically arrange in a particular way. Okay, so for example, if a cell will only divide in one plane and not typically, or maybe only typically stay stuck to each other for a little while, they might form pairs, otherwise known as diplococci. These are all round cells to begin with. Okay, or if it only divides in one plane and tends to stay stuck, if the cells stay stuck together for a long time, okay, then they can form chains. Okay, we see that sometimes up even in our micro lab. If the cells divide in two planes, you can see that right here, they tend to form packets of 4, which we refer to as tetrads. Or if they divide in 3 different planes, they form packets of 8 cells, kind of cube-shaped arrangements that we refer to as sarcina. Okay, let me see. I looked up, that's a Latin word for pack or bundle. All right, so it's really like a cube-shaped arrangement. In practice, it's kind of hard to tell the difference when you're looking at a 2D image, whether it's 4 cells there or 8. You don't have the right angle on it sometimes. So, okay. And then some cells will divide in basically any number of different planes and all stay stuck together. We just call those clusters. oftentimes they're referred to as grape-like clusters, actually. Okay, so there's a little terminology that you should become comfortable with in describing the cells that we see. Again, both their shape, their morphology, and how they tend to arrange. It's a little bit simpler when you talk about how bacilli arrange, because bacilli don't divide in lots of different planes. Basically, bacilli are always going to divide. They get longer and longer, and then they pinch off in the center and divide. Okay, so in other words, a bacilli would never divide like long ways. Okay, we just never see that happen. All right, so it's a little simpler. Sometimes you get just single bacilli. That's pretty common. Diplobacilli is when you have two that stay stuck together. Streptobacilli, that prefix strepto means chains. Streptococcus, round cells in chains. Okay, so those terms start to make sense a little bit, hopefully. It's a little bit less common for bacilli. Anthrax tends to form chains like that. Bacillus anthracis is an example. And the other thing that you see a little bit less common, or it's a little bit harder to recognize when it does happen, are these palisades. Okay, so that's when it almost looks like, oh, they're dividing longwise and then just staying stuck together. That's not what's happening here. They have some bacilli, when they divide, will stay attached at just one little spot, so they'll kind of hinge around. We call that snapping division. In other words, there's kind of a hinge. So they stay together just one spot, so they hinge around, and then when they keep doing that, they tend to stack together, what we call palisades. That term palisades came from like a fence around a fort. You know, they would cut a bunch of trees and stick them up, you know, side to side, and that's the palisade arrangement. So, and again, in a micrograph here, that's a little bit more difficult to actually recognize. All right, another really useful thing when you're trying to identify organisms or put them into groups is to decide whether the organism makes endospores. And we'll talk a little bit more about this structure later. If you are in lab, you will get a chance to do this endospore stain. The endospores, the ability to form endospores is quite limited. So I'm only going to say only a few genera are able to make endospores. It takes a whole bunch of unique genes in order to be able to do this. And what they are basically it's a survival mechanism. Okay, so the ability to sporulate, organisms will do that when they encounter some harsh environment. Okay, so nutrients are getting harder to find. I'm going to hunker down into a spore state where I'm not metabolically active anymore, and I can just persist until conditions improve. All right, so it's not a reproductive thing. In this picture here, the little green ovals, those are the free endospores. And then the red rods, these are the metabolically active cells. So we got a mix of both in this picture here. But you can see some cells where the spore is starting to form inside the cell. So you get one spore from one metabolically active cell. It's not reproductive. It's just a survival mechanism. So because only a few genera can actually do this, that makes those genera kind of unique. It makes sense to kind of group those organisms together that are able to form spores. All right. So we'll talk a little bit more about that spore cycle probably next time. Okay, so some of these simple criterion that we've been talking about, again, the morphology of the cell arrangement. Another really common thing to use to group organisms together is the Gram reaction. And we'll talk more about what that means for an organism to be Gram positive or Gram negative shortly, but it has to do with differences in their cell wall. It's a really useful thing. Let's see, whether they make endospores or not, we just talked about that. And the other thing that's kind of an easy thing to look at is the colony morphology. Okay, so the cell morphology, you need to have a microscope to actually see the cells. But the colonies, remember, this is what we see with the naked eye when we've got some bacteria growing on some solid media. Remember we talked about last time, oh, it's good, you want to get isolated colonies here, if possible, and then you can compare and see if those colonies look the same, to see if possibly it's all the same thing, if you have a pure culture or not. If you are in lab, you will get a little bit more practiced at looking at colonies. I didn't post this picture or this graphic because I don't want you to memorize all these words. I just want to show this to show the fact that, oh, there's a lot of different ways that we can describe those colonies. And one organism tends to always make colonies that look the same. Okay, so they can have a really kind of unusual shape to them. They can have a weird margin to the edge. Sometimes they're flat, sometimes they're kind of, popped up in the middle here, which is kind of cool. Different size colonies, they can be dry looking or glistening. Of course, there's lots of different colors. So there's a lot of different ways that we can describe colonies. And if you're working with one organism for a long period of time, you tend to get good at recognizing it. Like, oh, yep, I can tell just from looking at it, that's what that organism is. Okay, so that can be useful. So we're kind of, there's only so much you can accomplish with those things that you can just kind of see, though, in terms of categorizing organisms. We're relying more on biochemical things, and I'm kind of shifting back into chapter 4 here. So some of that, terminology about colony morphology and stuff is from chapter 11. Other things we use to put organisms in groups now, just for some examples, biochemical tests, like, can the organism utilize lactose as an energy source? Some organisms can and some can't. And this is a test, you'll be doing a lot of these kind of tests if you're in lab. But here's a test where we give organisms just lactose and see if they can use it when they ferment lactose. Remember I said lots of times when bacteria ferment a sugar, we get what kind of end products? Acidic, yes. So we got a pH indicator in there, yellow color change. Yep, that organism can use lactose. So it's one way to characterize organisms. There's actually a little tube in here, a little upside down tube that's used to catch gas. So you can see a gas bubble in this tube and not in either one of these. That just means the organism is using a particular fermentation pathway that will give gas as a byproduct. So that's one further way to characterize, give us a little more information about the organism. This is another, actually, a test for gas production, organism that can make hydrogen sulfide. We get a black color change there. Probably, we do some tests to see what kind of enzymes the organism can make. That will give us hints as to what group they should be in. Probably less often we would look at antibiotic sensitivity, because that can differ a lot just in within a species. Even we can have different strains of a species, variations of a species, but that's just sometimes helpful to characterize organisms. And even beyond biochemical tests, we're moving more towards, sequencing that ribosomal RNA, or doing serological tests where we're using antibodies to really look at, specific maybe proteins or, variations of species. So we're moving more into more molecular means. And just to kind of wrap this up, where do you go? Is there one source that you can go to find out all the characteristics of a particular organism? Burgess Manual. It's got about 5 different volumes to it. We have an old one of this up in the prep room in lab, but it's supposed to be sort of the go-to source if you want to know anything about a particular organism. It's sort of a consensus of what of scientists at any one time and what they think about particular organisms. So just thought I'd throw that in there. Okay. Right. Questions? Don't be afraid to jump in with any questions. All right, we're going to move on to chapter 3. We spend a good bit of time in this chapter. It's cell structure of both prokaryotic cells and eukaryotic cells. Every once in a while, I'll throw in the archaea, like if I happen to know about them, but we just don't spend as much time on the archaea because, as far as we know, none of them cause disease. So they get kind of short shift a little bit, but all right. So what kind of things we're going to be talking about? Difference between prokaryotic and eukaryotic cells, that should be reviewed. We're going to move through that pretty quickly, but we're going to run through a prokaryotic cell, and again, mostly focusing on bacteria here, looking at all the structures, and then we'll run through some eukaryotic cell structures and functions. I'm going to do that kind of quickly because really, if you are interested in organelles, you should probably take a cell biology course. I kind of just hit on a few that are going to be important for later on when we're talking about our immune cells and how they function. So trying to focus a little bit more on the prokaryotic structures, but in some cases, it's helpful to compare the prokaryotic versus the eukaryotic structure. Okay, so should be review difference between a prokaryotic cell and eukaryotic cell. So I didn't make you write this all again. Prokaryotes, right? They lack a membrane-bound nucleus, and they don't have any internal structures that are bound by a membrane. Okay, so they don't have Golgi apparatus, they don't have endoplasmic reticulum, they don't have lysosomes, any of those organelles. What size approximately are prokaryotic cells? They're usually less than or equal to about 1 micrometer. Okay, so that's kind of as a general rule, their size. Okay, so if you look at on the left here, these bacteria cells, okay, the units here, it's a scanning electron micrograph, about a micrometer, like if you're measuring a bacilli, it's going to be longer one way than the other. Okay, here on the right is a typical eukaryotic cell. Looks like a paramecium. Look at the units there, a lot bigger. All right, so if we look at a typical eukaryotic cell, of course they have a membrane-bound nucleus. They have membrane-bound organelles. We're talking about algae, fungi, plants, animals, protozoa. There is a difference between algae and plants. Algae are not just plants that live in the water. We'll mention that, but we're going to really focus a little bit more. I don't know if we'll get into it next time or maybe the time after that. A few fungi and a few protozoa, their characteristics. But eukaryotic cells are generally between about 10 micrometers. Micrometers, okay, and 100 micrometers. Okay, so at least 10 times bigger than a bacterial cell, kind of on average, all right? So big difference in size. All right, so like I said, we're gonna start from the outside of a bacterial cell and work our way in. So the first thing that we come to, most bacterial cells are going to have some kind of a glycocalyx. Okay, and this surrounds the outside of the cell, kind of a slimy substance that's secreted outside of the cell, typically made of polysaccharides. Okay, now there are a few exceptions there. Some cells, it's more protein based, but that's the exception. Most often they're polysaccharides. And that is important when we're thinking about staining cells and trying to see the glycocalyx, because these polysaccharides are not charged. And usually when we're using stains, the reason they stick is because unlike charges attract. So if you're trying to see something that doesn't have a charge, it becomes more difficult to actually stain it. Okay, so we'll get to that in a minute. But like I said, most bacterial cells produce some kind of glycocalyx, whether it's a really thick one or a really thin one, because it does have so many advantages to the bacterial cell. Okay, so three main functions: adherence, either to some kind of host cell or structure, or to other cells, other bacteria cells, like if it wants to make a biofilm, right? Lots of cells together in kind of community, they kind of stick together with this slimy glycocalyx. What kind of host structures might a bacterial cell stick to? Good examples in your mouth, right? You have probably on the order of 200 different bacterial species that secrete a glycocalyx to help them stay in your mouth so they can stick to your teeth instead of getting washed down into your digestive tract. Okay, so without that glycocalyx, they wouldn't be able to stay there, but that's what contributes to tooth decay and plaque formation, all those bacteria in your mouth. Another function, it makes the cell more resistant to drying out, to desiccation. Okay, a cell that, say, gets coughed out of our lungs and maybe lands, on some surface, it's not going to dry out as quickly if it has a glycocalyx around it to keep it moist. Okay, so it's going to be viable for longer, maybe out in the environment. Okay, and within a host, that glycocalyx will make the cell more resistant to being engulfed by our white blood cells. Okay, more resistant to phagocytosis. We'll just say white blood cells. Okay, our macrophages or our neutrophils that patrol around to get rid of invaders, they are going to have a harder time engulfing a bacterial cell that has a glycocalyx around it. Well, they have ways around that, but at least, sort of their first meeting. Okay, so pictures from your book. We typically will consider a glycocalyx as either a true capsule, meaning it's very thick and kind of, organized around the cell wall of that cell, or it's maybe a little bit thinner and less organized. Then we would call it just a slime layer. Okay, so really this is kind of a continuum. Again, thick glycocalyx, we would consider it a true capsule. It's kind of thinner, not bound as well to the cell. We might just call it a slime layer. Okay, so it's really common for cells to have somewhere on that continuum. I know that in lab, we have you stain two different organisms. One that makes a capsule, one that really sometimes makes a little bit of a slime layer. So sometimes it's not, sometimes it looks like they both have a capsule, but in reality, usually one has more than the other. Okay, so just an example there. And I may have posted this, I think I did post it, but when we do this in lab, usually our strategy is to stain the background, because you can see here, the background is red. The stain that we use kind of seeps through the capsule a little bit to stain the cell wall. But then this whole portion around those cells that's white, okay, the stain doesn't stick to it, that's the capsule. Okay, so we get this kind of halo effect when we do a special staining procedure that we don't see when we're just trying to stain the cell walls. So can be useful, kind of interesting anyway. Okay, so the next structure that we would come to, some organisms have a flagella. Okay, and I'll just point that out here. They're not present on all bacteria. Organisms that are round, as far as I know, don't have a flagella. Okay, so the purpose of the flagella is just so that the organism can move around, can move in a purposeful way towards some attractants, like some nutritional source or maybe sunlight. Okay, so they function in motility. Organisms that don't have a flagella are just at the mercy of their whatever medium that they're in. They're just going to have to float through the medium most often. Okay. So the structure of the flagella, and I'll note that the prokaryotic flagella, the bacterial flagella, is way different than the eukaryotic one. So we'll keep this in mind, and we'll kind of compare them when we talk about the eukaryotic one. But they're composed of a filament, a hook, and a basal body. Okay, so the basal body is what anchors the flagella into, we'll say, the cell wall, or sometimes to the cell membrane. Either one. There's a hook region then that kind of connects the two. And the filament is composed of the protein flagellin. Yeah, I love that when it kind of makes sense. And that name of that protein is kind of reminds you of the structure. So protein flagellin, just lots of different subunits of that stacked. Okay, so here's this figure from your book. It may be kind of tiny, hard to see if you printed this out. They show it two different ways here. They show it anchored in a gram-positive cell wall and a gram-negative cell wall, and we'll get to that cell wall structure in just a minute. I'm not too worried about where these rings are for the basal body, whether they're anchored, in the cell membrane down here or kind of within the cell wall. Whatever, the basal body anchors it to the actual cell. You've got this hook region right here, and then the filament. Okay, so it shows the motion here. It moves in a circular motion. Okay, so kind of spins around either one way or the other. Moves by rotation. Okay, the arrangement of flagella is different for different cells. So a little terminology here. If the organism, this on the top picture here, it's kind of hard to see those flagella. It's a scanning electron micrograph, but this has flagella all over the body of the cell. So I'm going to write this a little bit bigger, peritrichous. Okay, that prefix peri means all around. Okay, so if you're going to measure the perimeter of a room, you're going to measure all the way around the room. Okay, so trichus refers to like hair-like structures. So hair-like structures all the way around the cell. Okay, and a lot of bacteria do have a paratrichous flagella arrangement. We forget that because when we see bacteria in the lab, just staining them under our light microscopes, you can't see the flagella. They're too small to be seen. You have to do a special dye so that you kind of build up enough dye on each flagella that enables you to see them. And that's supposed to be pretty difficult to do. So it's easy to forget that a lot of organisms like E. coli, they've got flagella all around the cell, but some bacteria will just have a single flagella. We could call that monotrichous. A polar flagella, that means that it's one at one end or the other. So in other words, on a rod-shaped organism like this, you would never just have one flagella sticking out like from the side of its cell. Okay, we don't ever see that. It's either going to be at one end or the other, maybe both, but then it wouldn't be one flagella. Okay. It's also common to have a tuft of flagella, and I decided so there's a special like term to describe that that I decided that I don't need to fill your head up with some of these terms. So, but then I realized, oh, it's on your learning objectives. So there's a couple terms there for your learning objectives that we're not going to worry about. A tuft of flagella is technically called lophotrichous, and there's another term that we're just not going to worry about. So just know that there's different flagellar arrangements. Some organisms have one, a lot have, flagella all around their bodies. It's also possible to have kind of a tuft. Okay, so some variation there. And of course, there is this kind of oddball flagella called an axial filament, which remember the spirochetes have. Just kind of a close-up of that. This is a kind of flagella that's actually enclosed in a membrane. So an axial filament is correctly called an internal structure, where the other, like the paratrichous flagella, those are considered external because they're not enclosed in a membrane. Okay, so that's one difference. How does this axial filament actually get the organism to move in a corkscrew manner? There's lots of endoflagella. Okay, so many endoflagella. So all these little, see, this is the axial filament right here. And then there's lots of little filaments that are considered endoflagella, and those are actually what are spinning around, apparently. All right, and this is the organism pictured here, just in case you're interested, is Treponema pallidum. That's the organism that causes syphilis, right? So we think that because of that spiral motion, it can get into places maybe where other organisms wouldn't be able to get into. All right, so a little bit more about the motion of an organism that has flagella. You can kind of get lost on YouTube looking at pictures of bacteria, motile bacteria, or at least I have been. Like I said, bacterial flagella are always going to rotate, and it depends on which direction they go it depends on the direction of rotation as to how the organism moves. Okay, so for an organism that has a paratrichous arrangement, okay, if the flagella are moving counterclockwise, then the organism will move what we say a run. Okay, so in other words, when the flagella are moving counterclockwise, all of the flagella actually will coordinate, okay, kind of off in a clump, and then the organism will move in a very directed, a straight way. Okay, but if they're moving towards some attractant, it may realize, oh, I'm not going in the right direction. And so the flagella will start to spin the other direction. And when they are moving clockwise, then the organism will, what we say, it will tumble. Okay, so when the flagella move clockwise, all the flagella are kind of doing their own thing like this, and that causes the cell to kind of spin around and tumble around. Okay, so then when that stops and they coordinate again, it will head off into another direction. So it kind of will eventually end up, hopefully, towards that attractant. Okay, and there is one animation, I think, that's along with your homework, that's pretty decent in your book that kind of shows this. I have found other ones on YouTube that I think are good. Again, I wish I could just show them to you now. But when you get a minute at some point, this first one is just a cartoon animation that is kind of shows some runs and tumbles kind of nicely with some jazzy music. And then both of these, start them kind of, I think they're only like 3 minutes long, but if you start at like about 2 minutes, I think, for both of them, it's actually a camera on a light microscope where somebody has done just a wet mount. So that means they haven't stained the cells. That way they're alive. And again, if you dim it right down, if you dim it just right, you get good enough contrast that you can see them moving around. So you get to see in real time them kind of spinning around and moving off. And it's just kind of cool to see. I always try to do a wet mount as part of my lab, kind of at the end of the semester when you've gotten really good. It's hard to find your cells when you don't stain them, but by the end of the semester, we're practiced enough that we can usually find our cells and kind of watch them and see if they're moving in runs and tumbles. Oh, we know that they have flagella. If they're just kind of floating along, then they don't. So it's a way to test for motility, in other words. Okay, so other structures that we find outside of the cell. Fimbriae and pili. Both of these we typically see, and I know I haven't talked about gram-negative versus gram-positive yet, just bear with me. There may be an exception or two to this, as there usually is with any rule that we have, but for the most part, we think these structures are only in organisms that have a gram-negative cell wall. And they're very similar structures in that, in fact, sometimes we kind of, well, do you call it a fimbriae or you call it a pili? Sometimes we use those terms interchangeably, which is a little bit confusing. But typically, if we call them fimbriae, there's lots of them. So in this picture here, the really long things, those are flagella, but all these little short hair-like things around the cell, those we would typically call fimbriae. There's lots of them and they're short. The function of fimbriae is typically for attachment. Okay, so for example, E. coli has lots and lots of fimbriae so that it can stay attached to the endothelial cells of our large intestine. Okay, that's the cells that it gets nutrition from and what's passing, through our large intestine as well. But without those fimbriae, it would just get washed out of our body. So it needs those for attachment. Pili, typically there's only one or two on a cell when we call them pili. And the function here is for conjugation. And we're going to talk more about conjugation later, but conjugation is a kind of horizontal gene transfer. So two cells in the same generation, one is able to transfer genes to the other because of this pilus. Okay, and we now know that the DNA doesn't pass right through the pilus. Basically, the pilus just is a way to bring those two cells together, and then a pore opens up between them. Okay, so we're going to talk more about that later, but usually there's only one or two. The reason that we kind of mix these structures up a little bit is because the composition is the same of both pili and fimbriae. They're made of a protein called pilin. Okay, so whether you call it one or the other, we're not going to worry about that too much. And I noticed that your book, I think this error, I forget, I went back and checked to see, I keep checking to see if they've corrected this in the e-text. But this is definitely not a transmission electron micrograph on the right here. That's definitely showing the outside of the cells. Okay, if it was transmission, it would be a cross section. So just that's an error there. Okay. All right, so we're finally coming to the bacterial cell wall. Okay, and it seems like that we spent a lot of time on the outside of the cell, but there's not a lot actually inside of the cell to talk about. So don't worry that we're spending too much time outside there. Almost all bacterial cells have a cell wall. There's one genera that I know of that they don't have cell walls, but most bacteria do have cell walls. Why do they have a cell wall? Well, it helps maintain the shape of the cell. So for instance, if there's mutations then in the cell wall, then it can, with certain structures in it, we know that the cell can have a shape different than what it normally would. And it helps protect the cell from changes in osmotic concentration. So for example, if you put, I don't know if you've ever done this experiment, I think years ago we did it in Bio 120, but we put red blood cells in a wet mount, then we put like drops of pure water on one side and wicked it across and tried to see the red blood cells burst. Okay, because when they're in a hypotonic medium, they tend to take in water and burst. And I think last time when I was talking about osmosis, I might have gotten mixed up. I might have said that the wrong way. Nobody called me on it, but when I was going up to my office afterwards, I thought, oh, I think I said that wrong. Anyway, if you do the same thing with the bacterial cell, you put it, put some pure water. Okay, bacterial cells almost never burst because they have this strong cell wall that protects them in changing solute concentration. So that's an important thing. Composition of the cell wall. We're going to spend some time on that. Like I said, all bacterial cells that have a cell wall, they're going to have peptidoglycan in their cell wall. Whether they have a lot of it or a little, they're at least going to have some peptidoglycan. So what is that? It's a polymer of just what it sounds like. So the prefix there, peptido, that should make you think peptide. Okay, so in fact, it has some amino acid cross bridges. Okay, and then the second part of that word glycan, that should make you think sugars. Okay, there's actually two different sugars that repeat. Okay, and one of them, we abbreviate it NAG. That stands for N-acetylglucosamine. See if I can spell these correctly. Okay, and then the other sugar we abbreviate NAM, and that stands for N-acetylmuramic acid. Okay, so it's a compound with both sugars and amino acids. Okay, and this is your, so I didn't post this, but just so if you understand, if structures speak to you a little bit, both of those sugars are very similar in structure to glucose. So if you look at N-acetylglucosamine, the only difference here is at this second carbon. Otherwise, it's exactly the same as glucose. And N-acetylmuramic acid is different in two different carbons there. Only difference that it is from NAG is this orange part. So again, both of them similar in structure to glucose. So hopefully that helps you remember that they're sugars. And we think that this is the way those amino acids and those sugars kind of line up. All right, so alternating sugars, and then you have these amino acid cross bridges, both from row to row horizontally and kind of from row to row up and down. So I think a really good analogy here is a chain link fence. Okay, remember cell walls, yes, they protect the cell. They're not protective in that they don't let compounds in that would hurt the cell, right? That's the cell membrane's job to be selectively permeable. So the cell wall is not selectively permeable. Pretty much any molecule that's small enough to fit through there is going to go through, just like a chain link fence. But it does provide some rigidity and support. Okay. Now I want to just take a minute and go a little bit beyond the book. The book doesn't talk about how the cell grows, but I want to mention that now because we're going to come back to it later when we think about how to kill cells. Because anytime we talk about some compound that is unique to bacteria, then hopefully that makes us think, oh, magic bullets. Okay, I want to think about something that would kill bacteria cells and not be toxic to a human cell. Okay, that's basically what we hope antibiotics are. All right, and in fact, we have a lot of antibiotics, about half of the ones in use, that target bacterial peptidoglycan. Okay, because if it targets a structure that we don't have, it's much less likely to be toxic to us. We're always going to have side effects or allergies or things like that, but it's much more likely to be safe. Okay, so when a cell is growing is actually when it's the weakest, because in order to grow, it has to make that peptidoglycan shell larger. So it actually has to, cut bonds while it's inserting new subunits to make it larger. Okay, so a couple of enzymes that the cell needs to use. Autolysins, and again, this is not in your book. Autolysins are enzymes that cut the bonds between sugar units, between the NAG and the NAMs, and the amino acid cross bridges. Okay. And then there's another class of enzymes, and they have to make sure that that's done in a very controlled way. Those enzymes can't be making a bunch of cuts and leave the cell wall vulnerable. There's another class of enzymes called transpeptidases, and they seal the bonds back up in the amino acid cross bridges. Okay, so you have to coordinate the amounts of those enzymes produced. Okay, so this is the best graphic that I could come up with here to show how this works. So the new NAMs and NAGs are going to be made inside of the cell. Okay, this is cytoplasm, this is inside. And then they have to be transported through the membrane, a special channel there we're not going to worry about. So, and then these bonds, some of these broken so that the new subunits can be put in there. Okay, in the process, some of these amino acid cross bridges have to be broken apart and then resealed with the TP, the transpeptidases. Guess what antibiotics targets, or many antibiotics like penicillin, they bind to the transpeptidases so that they can't reseal those links. Okay, and then that weakens the cell wall, and the cell can actually burst due to osmotic pressure. So that's what this little clip shows. It's a time-lapse video that shows some bacteria cells in the presence of penicillin and how they grow, and then all of a sudden they burst. So take a look at that. We'll pick it up there on Thursday. Have a good day. It's gonna rain, maybe storm. So get to where you want to go.